Hello, my dear friends. Welcome to LivePrep. Myself, Dr. Gajapati Varma. On 4th May 2025, NTA has conducted NEET UG examination. So immediately after the examination, so many institutes have released their answer keys. So here, we are mainly focusing on the biology only. So here in this particular video, I will be explaining the answers for each and every question that was given in the NEET 2025 biology section only. Okay, here we are discussing only about the biology section. The thing is, so even though majority of the institutes are giving almost the same answers, but for few questions, some students are still having some doubts. So that's why here I am going with, anyhow, I am going with all the questions, but I will be focusing on those questions where exactly the students are having the doubts. Okay. But here what I did is, so I have segregated all the 90 questions into different chapter wise questions. So in this way here, what I will do is, I will go in a particular NCRT sequence wise. Okay. Whatever the chapters, NCRT chapters are there in that sequence. So all these 90 questions of NEET examinations I have distributed. So accordingly, we will be going with these particular questions. And for some of the questions, they are direct answers. And for that, I am not giving any explanation. So they are very simple. So there I am not wasting your time. But for some questions, so they need some explanation. So at that particular location, definitely I will give little bit more explanation. Clear? Now let us quickly go with this one. And anyhow, so already I made a separate video regarding the analysis of this particular NEET 2025 biology paper specifically. Okay. So from each chapter, how many questions were asked, and that too, what type of questions they have asked. So in this way, I made a separate video. So please go through that one also. So you will get some basic idea regarding that one. So in this way, so anyhow, please remember, as per NCRT, there is no botany zoology separately. Okay, for our convenience, we are classifying this botany and zoology into different things. That's it. The thing is, so in some locations, some chapters they are considering under botany, the same chapters in some other location they are considering under geology. So in this way, some interchanges are also taking place. So that's why there is no exact clear-cut differentiation here. So that you cannot say that, so these are the only botany chapters, these are the geology chapters. Okay, because some chapters are like, so basic zoology related, so that you can understand. Some are basic botany related, that we can understand. But some chapters are common chapters, general chapters, that you can consider under anywhere. But here what we are doing is, for the understanding purpose, so generally these are the chapters that we are considering under zoology, these are the chapters that we are considering under the botany. So in this way, for this NEET 2025 biology, if you are giving the weightage, so here out of 90, 49 questions they have asked from the botany part, and 41 questions they have asked from zoology part. Anyhow, these things also we have discussed there. And here class 11th, total 39 questions they have asked. From class 12th, total 51 questions they have asked. In this way, they made total 90 questions for the entire biology here. And out of this, direct questions are 41 questions, and assertion reason questions are 8, and statement 1 and 2. Statement 1, statement 2, like that total they have given 8 questions, and match the columns. Column 1, column 2, match the following, right? So these questions they have given total 11 questions, and a diagram based total 4 questions, means including pedigree and all. So I have considered it as total 4 questions. And then multiple statements. So here they will give some list, there may be some statements or there may be some options they will give, and out of the how many are correct, likewise we need to select from that. So that type of questions are approximately 18 questions. Okay, and directly whether they are correct or incorrect, even in this multiple statements also, majority of the questions are again identify the correct, likewise they have given. Okay, so I have, since there are multiple statements like, or multiple some list they have given, so that I have considered separately. But there are actually 3 questions directly they have asked, like identify the correct statement, identify the incorrect statement, likewise they have asked. Likewise, total 3 are there in that. One is for identification of correct, and 2 are for identification of the incorrect statements. So in this way, overall 90 questions were given. And anyhow, with a more elaborate explanation, I have given in a separate video. So please go through that. Okay. If you are not going through that, so here you will get the some basic idea. Now let us quickly go with each and every question. But please remember, here I have classified this as per the NCRT chapters. Okay. So in the NCRT first chapter is the living world, but here from the living world there are no questions. Okay. So now directly we are going with the biological classification. Okay. So from the biological class, what are the questions here? So here, So here they are saying that each of the following characteristics represent a kingdom proposed by the Whittaker. Arrange the following in increasing order of the complexity of body organization. Very, very simple. Direct, these are NCRT statements. So simply you need to arrange them. Okay. Anyhow, since it is simple, I am not going for the explanation. So answer is option 4. Clear? Next one. Which one of the following is the characteristic feature of the gymnosperms? See, before going with this one, so some students are expecting that, so see, in different locations, different sets they have released, right? So for each and every set, it will be a little bit difficult to make the answer key. So that's why here I have segregated. Anyhow, these are the questions, these are the options, and everything are these only, but jumbling will be taking place. So that's why don't worry about the set. So go through the question and answer accordingly. In your question paper, you just check it. Okay, so that will be much better for you. Next one. So this is from plant kingdom. So which one of the following is the characteristic feature of the gymnosperms? The very most important feature is seeds are naked. Okay, direct answer. Okay, next one. This is also from the plant kingdom. So here they have given the list A and the list B, or list 1 and 2. So directly here some uh, groups they have given, and then some examples they have given. Direct NCRT examples here. Okay, so for this the answer is option 4. For this type of questions I am not going with explanation. So directly we will go with the next one. Next one is also from the plant kingdom. In the biophytes, the gemme helps in which of the following? So what is the advantage of these things? They are helpful in sexual reproduction. So answer is option 4 here. Next one is also from the plant kingdom. Given below are the stages in the life cycle of the pteridophytes. So for the pteridophyte life cycle, they have given some sequence and we need to arrange them. Okay. So already we know the cycle. So as per the cycle, we need to arrange them. So for this one, answer is option 3 here. Okay. So those who are still having any doubt, you can pause the video and then you can check all these things. Clear? Because explanation is not, and this is the direct standard sequence. So that's why directly we are going with these answers. Next one. This is also from the plant kingdom. The correct sequence of events in the life cycle of bryophytes. Previous question is for pteridophytes. This is for the bryophytes. Here also there is a standard sequence is there. You please follow the sequence. And here the answer is option 1. Okay, next one. This is from the animal kingdom. And here, given below are the two statements. Assertion and reason they have given. Now let us see. Assertion. All vertebrates are chordates, but all chordates are not vertebrates. This is correct. Next one, reason. The members of a subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by cartilaginous or bony vertebral column in the adults. Correct. So both the statements are correct, and reason is the correct explanation for assertion. And these are the direct things that were very clearly mentioned in the NCRT. Okay. So here answer is option 3. Next one, and again from animal kingdom. All living members of the class Cyclostomata are. All the living members of the Cyclostomata, what are these? These are ectoparasites, mostly on the fishes. Yes or no? So they are ectoparasites. Option is on option 2. Answer is option 2. Next one, another from, and again from the animal kingdom. So what is the role of this water vascular system in the echinodonts? Direct statement from the NCRT. What the answer? Respiration and locomotion. Not only that, that is also involving in capture and transport of the food. Direct statements from the NCRT. So include these two. So A and C. Where is this one? Option 4. So it is answer is option 4. Next one, again from the animal kingdom. So while trying to find out the characteristics of a newly found animal, a researcher did the histology of the adult animal and observed a cavity with the presence of mesodermal tissue towards the body wall. Mesodermal tissue is present towards the body wall, but it is not present towards the alimentary canal. Okay, body wall, but no mesodermal tissue was observed towards the alimentary canal. So what type of coelom it is? It will be pseudocoelom. So that's why the animal will be pseudocoelomate animal. Yes or no? So answer will be option 4. Next one, morphology of the flowering plants. So here, so which of the following is an example for zygomorphic flower? So direct NCRT example it is. It is the pea plant flower. Yes or no? Next one. Again from the morphology of the flowering plants. Here they have given two statements. Here they have given the floral formula symbol they have given. Okay. Here this one they have given another one. In both the statements they give these two only. But whether they are giving these symbols for the corresponding words or not, that we need to observe this. What is the symbol of this particular word? Is it for zygomorphic or actinomorphic? That is for actinomorphic. And what is this particular symbol? It is for inferior ovary or superior ovary? It is for superior ovary. Okay. Simply these symbols they kept same in both the statements, but these words they have changed. So now which one is the correct one here? So statement 1 is wrong. Statement 2 is correct. So where is this one? It is in the option 2. Clear? Next one. Next one, this time it is from the anatomy of the flowering plants. So find the statement that is not correct, means incorrect statement they are asking with regard to the structure of the monopod stem. So go through the options here. So here in the option 3, what they give, hypodermis is parenchymatous. This is wrong. In the case of monopod stem, hypodermis is not parenchymatous, it is sclerenchymatous. So that's why it is the wrong statement. So answer will be option 3. Next one. So this time it is from the structural organization in animals. This is very, very important. In general, for the structural organization in animals, we expect only one question. Okay. But here they have asked 4 questions, and all the 4 questions are from frog only. Only from the frog they have asked. Now let us say this. In frog, the renal portal system is special venous connection that acts to link. So here regarding the portal system, they have discussed. Already in the regular class also we have discussed. Total 3 portal systems are present. Renal portal system, hepatic portal system, and hypophyseal portal system. Yes or no? Hypophyseal and hepatic portal system, these are present in the frog, even in the human also. But renal portal system, it is not present in the human, it is present only in the frog. So that renal portal system, directly they are asking. Renal means what? Kidney. Yes or no? Already regarding the portal system, very clearly we discussed. The name we are giving with respect to the collecting organ. Kidney is collecting the blood. So that's why it is the renal portal system. So from where? From the lower parts of the body, the kidney is connecting. So what are involved here? Kidney and the lower parts of the body. And this is applicable only for the frog. It is not applicable to the human. Please observe this. Clear? This is what answer is option 2. Next one. Again from the frog only. Frogs respire in water by skin and buccal cavity. Observe this carefully. Frogs respire in water by skin, correct. But what about buccal cavity? No, it is not by buccal cavity. Please observe this. So that's why respire in water by skin and buccal cavity. So this will be wrong statement. Observe carefully. And on land, skin, buccal cavity, and lungs. This is correct. So with respect to land, the statement is correct. But with respect to the water, the statement is wrong. So where is this? The statement is false for the water, but it is true for the land. Yes. So this is the option that we need to select. Clear. When it is present in the water. Please remember, skin is the main respiratory organ. On the land, all the 3 can be working. All the 3 are working. Observe carefully. Next one, again from the frog only. So what is the name of the blood vessel that carries deoxygenated blood from the body? From the body, deoxygenated blood should be collected so that it is going to the heart. So what is that one? It is the vena cava, inferior vena cava, superior vena cava. So here they have not given those many names. Directly they have given vena cava. So answer will be vena cava. Clear? This is what. Next one. Again from the frog only. So which of the following statements is correct about the location of male frog copulatory pad? So these copulatory pads are present in the males, and what is the location? So it is present on the first digit of the forelimb. For the forelimbs, on the first digit, this copulatory pad is present. Clear? So answer is option 2. We will go with the next one, that is cell, the unit of life. So here we will go with the question first. So here they have given about the assertion region. First let us see the assertion. The primary function of the Golgi apparatus is to package the materials made by the endoplasmic reticulum and deliver it to the intracellular targets and outside the cell. Yes, it is correct. Reason. Vesicles containing materials made by the endoplasmic reticulum fuse with the cis phase of Golgi apparatus, and they are modified and released from the trans phase of the Golgi apparatus. So in this way only now this packaging is taking place and the delivery is taking place. So that's why both the statements are true, and R is the correct explanation for A. Okay, this is what. Next one, again from the cell, the unit of life. From the statements given below, they have given some statements, so we need to identify the correct statements. The eukaryotic ribosomes are 80s and prokaryotic ribosomes are 70s. Correct. Each ribosome has 2 subunits, correct, larger and smaller subunits. The 2 subunits of 80S ribosome are 60 and 40, whereas that of 70S are 50 and 30, correct. And here they have given that for 80, 60 and 20, this is wrong. For 70, it is 50, 20, this is wrong. And 2 subunits of 80 are 60 and 30, this is wrong. For 70, 50 and 30 are wrong. Please observe this one. Okay. So 50, 30, even though it is correct, this part it is wrong. So that's why now overall if you see, up to ABC they are correct. D and E are wrong. So here they are asking about correct selecting the correct one they are asking, right? So what are the correct ones? Only A, B, C are correct. So answer will be option 3. Clear. Next one. Again from the cell, the unit of life. A specialized membranous structure in the prokaryotic cell which helps in the cell wall formation, DNA replication, respiration. What is that one? It is the mesosome. Clear? So it is the mesosome. Already in the NCERT we have very clearly studied. Next one again from the cell, the unit of life. Here they have given about the matching, direct matching on some structures they have given and their functions, locations likewise they have given. So very direct matching only. So answer will be option 2. Next one. So this time it is from the biomolecules. So from the biomolecules also huge number of questions they have given. Now let us see. The protein portion of an enzyme is called as, there are some enzymes they are called as simple enzymes. Some enzymes are called as conjugate enzymes or whole enzymes. So in the simple enzyme only protein is there. So there is no problem. But in the case of conjugate enzyme, protein part, non-protein part is there. Non-protein part is called as cofactor. Whereas the protein part is called as, so this is what they are asking, protein part of an enzyme. So mainly with respect to conjugate enzymes they are asking. So what is the protein part of the conjugate enzyme? It is called as apoenzyme. Clear? Next one again from the biomolecules. They have given some structures and now the names they have given what type of molecule it is we need to identify. So it is very simple what I will tell you. Adenosine. What is adenosine? Adenine plus ribose is adenosine. So adenine plus ribose means it is nucleoside. So please observe carefully. Sorry. Next adenylic acid. What is this adenylic acid? Adenine plus ribose plus uh, phosphate group. So 3 are there. 3 are there means it will be a nucleotide. Yes or no? Next one. Adenine. Adenine is directly a nitrogenous base. It is a nitrogenous base. Whereas alanine is amino acid. Okay. So that's why in the regular class I told you to remember these names. How exactly we are deriving these names. If you know this, you can easily understand this particular one. So answer is option 1. Next one. Which one of the following statements refers to reductionist biology? Very clearly, repeatedly in the regular class, I told to each and every student that even this unit introduction is also very, very important. This is the direct statement from the unit introduction. So if you are opening the cell, the unit of life chapter, before to that, there is a unit introduction. In that unit introduction, very clearly they have given. Okay, even before this, they have given explanation also. In the regular class, I have very clearly explained what is this reductionist biology, why it is called like this, and how, in what way this particular sentence is linked with the biomolecules chapter. Everything we have very clearly studied. Now you just see this physico-chemical approach to study and understand living organisms is called as reductionist biology. The same thing they have given. What is this reductionist biology? It is the physico-chemical approach to study and understand living organisms. Direct statement. Okay, many students are thinking that it is out of NCERT because you have never studied about this unit introduction. So very important. From the unit introduction also it is very important. Just in the next page of the unit introduction, sometimes regarding a particular process, sometimes regarding a particular scientist also they are giving. In the same paper regarding Ramdev Mishra. So that is also given just behind that unit introduction with respect to the ecology chapter. Yes or no? That was also given. So please remember anything that is printed in the NCERT textbook, whether it is present in the NCERT particular chapter in the unit introduction or in the exercise, where exactly it is, even from the QR code content also. There is a pedigree and that pedigree content is related to QR code content. I will show you all the things. Okay. So you need to go through each and everything related to NCERT. So then you can easily understand all these things. So I hope you can understand this. This is what the answer is. Next one. Again from the biomolecules. So which one of the following enzymes contain heme as the prosthetic group? So under the separate heading that is cofactors, we studied about the prosthetic group and then coenzymes, metal cofactors likewise, right? So here they are asking about the prosthetic group heme containing enzyme. What is that one? Direct NCERT example, catalase enzyme. Okay. And next one regarding the enzyme classification from there also. In the NCERT, they have given this one regarding the transferases. This is the substrate to this already a group other than hydrogen is attached. And when another substrate is also present here. So now there is a transferase taking place. Now this G is already attached to one substrate. Now this G is moving from this substrate to another substrate. This is what the NCERT reaction. And here in the NCERT reaction for this, yes, they have given some dash like symbol or prime like symbol they have given here. But instead of that, here they have given this asterisk symbol. That's it. And remaining reaction, everything are exactly same. The process, everything, even the explanation also they have given what is G, what is S, what is this S with asterisk. Everything they have given as it is, right? So what is this what is the enzyme that is involving in the catalysis of this particular reaction? It is transferase. So transferase enzyme. Okay. Next one. With that, the questions from biomolecules are completed. Now we are going in the next one, that is cell cycle and cell division. Only single question they have asked here. What is the main function of the spindle fibers during mitosis? What is the function of the spindle fibers here? So they are helping in the separation of the chromosomes. Yes or no? The same thing. Okay. Next one. So that's it. Next one. Next question is from the photosynthesis in higher plants. Now here, which of the following statements about Rubisco enzyme is true? What is the Rubisco enzyme full form? RUBP. What is RUBP? Ribulose 1,5 bisphosphate. This is Ruby. RUBP. And continue here. Ribulose 1,5 bisphosphate carboxylase oxygenase. Okay, it has two functions, right? So regarding that particular enzyme, they are asking about this one. It is an enzyme that is involving in the photolysis of water. No, and it catalyzes the carboxylation. What is this C? C is the carboxylation, right? Yes. So carboxylation of RUBP, correct. Okay. Carboxylation, oxygenation. So in this way, carboxylation is correct. So option 2 is correct. Anyhow, we will see the remaining. Active only in the dark? No. It is active in both dark and light conditions also. Next one. It has higher affinity for oxygen than carbon dioxide. No, it should be reverse. Okay. So anyhow, from the given statements, only this one is the correct. Okay. Next one. Next one from the photosynthesis again, and here they have given some chlorophyll and different pigment molecules they have given. What is the corresponding color? Direct matching here. Okay, there is no confusion here. Answer is option 4. Next one, regarding the respiration in the plants chapter, and here they have asked one question, and with respect to the complex 2 they are talking here. So what this complex 2 is doing, it is converting succinate to fumarate. And what is the name of the enzyme that is present here? Succinate dehydrogenase. Yes or no? So when we are studying about the Krebs cycle, so Krebs cycle generally we are saying that it is occurring in the mitochondrial matrix, but the one and only enzyme that is present in the inner mitochondrial membrane and that is related to Krebs cycle is succinate dehydrogenase. That is nothing but the complex 2 of electron transport system or electron transport chain. So what is this complex 2? It is the succinate dehydrogenase. Clear? So answer is option 4. Next one, plant growth and development. And here they have asked two questions. Let us see here. And here they have given some properties. And here they are saying that read the following statements on plant growth and development. And here we need to identify the correct statements. Let us see here. Parthenocarpy can be induced by the auxins. Yes or no? Yes, it is true. Next one, plant growth regulator. Please remember, regulator is a general term. Okay, they can help or they can also inhibit. This is a general term. So please observe this. Plant growth regulators can be involved in the promotion as well as inhibition of the growth. Since it is a general term, yes, this statement is also correct. Next one, dedifferentiation is a prerequisite for redifferentiation. Yes, this is also correct. Next one, abscisic acid is a plant growth promoter. It is not a promoter. So that's why this statement is wrong. Next one, apical dominance. Apical dominance promotes the growth of the lateral buds. No, when apical dominance is there, lateral buds growth is not promoted. So that's why what happens? D and E are wrong. A, B, C are correct. Okay. So choose the option with all the correct statements. What is that one? It is option 3. That is having A, B, C statements. So because these 3 are correct. Clear? Next one. Again from the plant growth and development. Which one of the following phytohormones promote nutrient mobilization which helps in the delay of leaf senescence in the plants? What is the hormone? It is the cytokine. The right option. Okay. Next one. Now from the human physiology, let us see here. From the breathing and exchange of gases. But actually, even though I have kept it under breathing and exchange of the gases, but this entire question is not from this particular one. If you see this emphysema, breathing and exchange of gases, angina pectoris, okay, this is from the body fluids in circulation, and glomerulonephritis, this is from the excretory products and their elimination, and the tetany, it is from the locomotion and movement. From 1 chapter 1 under the diseases part, at the end of this each of these chapters they have given 1 they have given here, and the corresponding conditions also they have given, and these are also direct NCERT content only. They are not that much confusing, but anyhow, so since the first one is related to breathing and exchange of gases, so I kept it under this particular one, and the answer for this one is option 2. Okay, direct and simple matching only. Okay, next one, from the body fluids in circulation they have asked one question. Let us see this. Cardiac activities of the heart are regulated by, so at the end of the chapter they have given the regulation, right? So there they are talking here nodal tissue. Okay, so here what happens in the case of heart, so it is myogenic. So these contractions, everything, they are generated from the muscle cells, specialized muscle cells called nodal tissue that is present within the heart itself. So it is correct. And not only this, even the sympathetic, parasympathetic nervous system, some content also they have given in NCERT, So regarding that, they are talking here a special neural center in the medulla oblongata of the brain. Yes, this is also correct. That is also having some ability to control it. Next one, adrenal medullary hormones. What are these? Epinephrine, norepinephrine. Yes or no? They are also able to control this cardiac activity. So that's why these three can regulate the heart activity. Whereas adrenal cortical hormones, no. So that's why what we need? ABC. Where is this ABC? So it is the option 3. Clear? So this is what the answer is. Next one is from the excretory products and their elimination. And here they have given the diagram. And this left side part for all these options is with respect to the proximal convoluted tubule PCT, and the right side things are for distal convoluted tubule DCT. Okay, and here in the NCERT they have given the diagram also, right? Now see this one. This is the tube, right? And these upper side arrow marks. Now these are absorbed from this particular lumen. These things are absorbed into the body, means medullary interstitium, and these arrow marks which are facing towards the lumen. So what are these indicate? So these are secreted by the medullary cells into the lumen. This is what the meaning that we need to observe. And now we know the basic concept related to this particular one, and based on that, if you see, so this is what the correct diagram. This is what the correct diagrammatic representation here. Yes or no? Bicarbonate, NaCl, water, all these are absorbed in the PCT. H plus ammonia are secreted into the PCT lumen. Yes or no? And now with respect to DCT, NaCl, water, bicarbonates are absorbed. And next one. So it is K plus H plus, if they are present excess, now they will be secreted into the lumen of the DCT. Yes or no? So now this is the one that is correct. So with respect to this particular diagram, option 3 is the correct. Clear? Next one. Next one is regarding chemical coordination and integration. From this particular chapter, they have asked total 4 questions. So they have given high weightage in this particular exam for this particular chapter. And here they have given some hormones and their locations and which one is secreting this. These are the direct examples that they have given. Answer is option 3. Next one. Again from the chemical coordination chapter only. And here they have given some properties of this medullary adrenal medullary hormones, and we need to identify whether how many are correct and wrong, like this. Okay, so choose the following statements regarding the function of adrenal medullary hormones. What are these adrenal medullary hormones? These are the flight or fight hormones, right? So yes, epinephrine, norepinephrine. Regarding that, they are saying it causes pupillary constriction. Will they cause pupillary constriction or dilation? Pupillary dilation they will cause, but here they give pupillary constriction, so it is the wrong statement. Next one, it is a hyperglycemic hormone. Hyper means increasing, glycemic glucose, they are helping in increasing blood glucose levels. In the NCERT, did they directly use this particular sentence? No, but what they said, these hormones help in breakdown of glycogen. So what happens because of the breakdown of glycogen? Because of the breakdown of glycogen, glucose will be released into the blood, so ultimately it will result in hyperglycemic condition. So these adrenal medullary hormones are can be acting as hyperglycemic hormones. True or false? It is true. A is false, but B is true. Next one. It causes piloerection. Direct answer statement. This is correct. It increases the strength of heart contraction. This is also direct answer statement. Correct. So what are correct here? B, C, D are correct. So B, C, D are correct. So answer is option 4. Clear? Next one. Which of the following hormones released from the pituitary is actually synthesized with the hypothalamus? With respect to pituitary, we studied no? Posterior pituitary neurohypophysis. Yes or no? It is secreting actually or releasing actually 2 hormones. What are those? Oxytocin, vasopressin. Vasopressin is also called as antidiuretic hormone. Actually, these hormones are synthesized by the hypothalamus, and they are stored in the posterior pituitary. From the posterior pituitary, they are released. And now, what are these hormones? They are asking. They have given some examples. What we need, what we are expecting, oxytocin or vasopressin we are expecting. So vasopressin is also called as antidiuretic hormone. So that option is directly there here. So answer is option 4. Next one. Again matching they have given here. So different organs they have given, and what are the different with respect to what are the different hormones that are secreted from this. Direct as it is information from the NCERT only. So answer is option 2. Clear? Next one. So with that, class 11th all the chapters are completed. Now we are going with the class 12th chapters. And now here first we are starting the sexual reproduction in the flowering plants. And here first they have given about this assertion region. A typical unfertilized angiosperm embryo sac at maturity is 8 nucleated 7 cell. Yes or no? Yes, it is 8 nucleated 7 cell. It is true. Next one. Next one, reason. The egg apparatus has two polar nuclei. Egg apparatus. So what are present in the egg apparatus? In the egg apparatus, one egg cell is present and two synergids are present. That's it. And these are the polar nuclei. Polar nuclei are not part of the egg apparatus. So what is the correct answer here? A is true, R is false. This is the correct answer for this particular question. Next one. So they have given some assertion region. Now we need to observe this carefully. Both wind and water pollinated flowers are not very colorful and do not produce the nectar. Yes, correct. Water pollinated and wind pollinated. If they have to attract the insects, then all these things will be there. But here insects are not playing role here, and it is by the water and wind. So that's why they are colorless. So almost white in color majority of this, and they are not producing the nectar. This is a correct statement. Next one. The flowers produce enormous amount of pollen grains in wind and water pollinated flowers because there is a huge wastage will be there. To compensate that particular wastage, they are producing huge number of pollen grains here. Now individually if you see, both the statements are correct. A is true, R is also true, but R is not the correct explanation here. Here what we are expecting, colorless, and why nectar is not produced. For that we are expecting the explanation. Okay, so that's why what we are. So what is the ultimate answer? Both A and R are true, but R is not the correct explanation for the A. Okay, this is what the answer is. Next one, again from the same chapter. How many meiotic and mitotic divisions need to occur for the development of mature female gametophyte from the megaspore mother cell in the angiosperm plant? So initially this angiosperm. So here what happens? This megaspore mother cell, this is the diploid cell that is undergoing the meiosis. So one meiosis. And after meiosis, 4 cells are produced, 3 will be degenerated and 1 will be there. And that 1 will undergo 3 mitotic divisions. So that 8 cells will be formed. Yes or no? But later on some fusions and everything will happen. So in this way, ultimately, this particular gametophyte will be produced. So in this way, here in this question, our focus is based only on the number of divisions. So here initially 1 meiosis and later on 3 mitosis will be taking place. Clear? So that's the answer will be 1 meiosis and 3 mitosis. That's it. Next one. Next one from the same chapter only, but this time it is again assertion region. Cells of the tapetum possess a dense cytoplasm and generally have more than one nucleus. Generally, we are observing the multinucleated condition. So it is correct one. Next one, reason. Presence of more than one nucleus in the tapetum increases the efficiency of nourishing the developing microspore mother cells. Yes, it is the reason. This is the very important reason. So that's why both the statements are correct and R is the correct explanation for A. Okay, observe carefully. Next one, from the same chapter. In the seeds of the cereals, the outer covering of endosperm separates the embryo by a protein-rich layer that is called as ellurone layer. Clear, direct NCERT statement. Next one. Next one from the same chapter again. So here they have given the matching, and this matching also again directly it is very important and direct NCERT based only. So answer will be option 3. Next one. So with that chapter is completed. Now we are talking about the questions from human reproduction chapter. The first menstruation is called as menarche. Okay, next one from the same chapter. Find the correct statement. They have given some statements we need to identify. So what are the correct statements? Let us see here. In sorry, in human pregnancy, the major organ systems are formed at the end of 12 weeks. Same statement also they have given here, but they have given at the end of 8 weeks. So definitely one of this only will be correct. So which one is the correct? If you see the NCERT, you can understand this. It is the option A that is correct. So option B is wrong. Okay, next one. In human pregnancy, heart is formed after 1 month of gestation. So with respect to this afterward, there is a small confusion. So generally, this heart will be formed in the 3 weeks, approximately in 3 weeks. 3 weeks it will be formed, but heartbeat will start in 5 to 6 weeks. Okay, but whatever it is overall, if you say around 1 month, if you say so, but here as per the given options, if you say, you need to consider that this is a correct statement. So here A is correct, B is wrong, C is correct. Clear? Next one. In human pregnancy, limbs and digits develop by the end of the second month. This is correct. In human pregnancy, the appearance of hair usually observed in the fifth month. This is also correct. So what is wrong? Only B is wrong. Remaining all are correct. So A, C, D, E. Remaining all these four are correct. So answer is option 2. Clear? Next one. Again from the human reproduction chapter, they have given the matching with respect to the sperm cell parts and their functions. They have given direct information, no need to confuse here. So answer is option 3. Clear? Next one is again from the human reproduction. Now let us see here. Here they have given some statements. We need to observe this carefully. The reproductive division for the human female gametogenesis starts earlier than that of the male gametogenesis. True or false? It is true. Because in the case of human female, the gametogenesis is occurring in the fetal life itself. Yes or no? But in the case of male, what happens? At the puberty it will begin. So that's why this statement is correct. Next one. The gap between the first meiotic and the second meiotic divisions is much shorter for the males when compared to females. Is it true or false? This is also true. In the case of males, there is no stoppage. Once these meiotic divisions are starting, continuously happening. Meiosis 1, meiosis 2 will be completed. But in the case of female, what happens? Meiosis 1 is starting in the fetal life, and there it will be arrested. And again, some of these cells in the menstrual cycle after the puberty. So once puberty is attained, so then in the menstrual cycle, for every menstrual cycle, only few cells are again continuing, and remaining are still in that meiosis 1 only. Yes or no? And in this way they are going. Suppose those which are entering into this particular menstrual cycle, now they are slowly developing, and during this phase some of them will be undergoing means they will be reduced, and only few are trying to develop, and ultimately only one ovum is released. Yes or no? So even though we are saying the ovum, actually it is the secondary oocyte that is released from the Graafian follicle. And if you clearly observe that when this Graafian follicle is released, and in that Graafian follicle, sorry, in when the from the Graafian follicle, when the secondary oocyte is released, in that secondary oocyte again meiosis 2 is arrested. So in this way, it is stopping and then resuming, stopping, resuming. Likewise, it is happening for the female. But these steps are not applicable to the male. In the male, they are happening continuously. And should or not. So that's why even this statement B is also correct. So A and B are correct. Next one. The first polar body is associated with the formation of primary oocyte or secondary oocyte. Technically, it is with secondary oocyte, but here they give primary oocyte. That's why it is wrong statement. So C is wrong. Next one. Luteinizing hormone surge means LH surge leads to disintegration of the endometrium and onset of menstrual bleeding. No. LH surge is related with what? Ovulation process. So that's why C and D are wrong. A and B are correct. So A and B are true. Next one. Again, from the human reproduction. Twins are born to a family that lives next door to you. The twins are baby and girl. So please observe carefully. Twins are there, and one is boy, one is girl. That means, are they monozygotic twins or dizygotic twins? They are dizygotic twins. They are developed from two different zygotes. Understood or not? So that type of twins are also called as a fraternal twins. Dizygotic twins are also called as a fraternal twins. So answer is option 4. Clear? Next one, from the reproductive health. Here they have given some statements, but please remember some statements are not from NCERT. So you need to observe the options and then accordingly you need to take the decision. And here what they're saying, what are the potential drawbacks in adoption of IVF method? With respect to IVF method, what are the drawbacks? What they are saying? High fatality risk to mother. No, there is no risk to the mother. Next one, expensive instruments and reagents. Yes, they are expensive. This statement they have given in NCERT. Next one, husband and wife necessary for being the donors. So they don't need to be the donors. So someone else can be the donor also. So that's why this statement is again wrong. Next one, less adoption of the orphans. Yes. So now if there is no IVF method, those couples who are not having the children, they may go for adopting the orphans. But if they are going for the IVF, the chances of adopting an orphan will be decreased. So that's why less adoption of orphans is a correct statement. Okay, next one. Not available in India. This is wrong. It is available in each and every city of India. Okay, next one. Possibility that the early embryo does not survive. Yes, this is correct. So what are correct with respect to this one? IVF. Expensive instrument and reagents, less adoption of orphans, and then possibility that the early embryo does not survive. So B, DF, so BDF. So these are the statements that are related to the given question. Clear? Next one. Next one, from the principles of inheritance and variation, they asked only 3 questions. Let us see this. What is the pattern of inheritance for polygenic trait? So polygenic trait will come under non-Mendelian genetics. Please remember here, we studied about monohybrid cross, dihybrid cross, like this, right? And these crosses, they are Mendelian, they are following the Mendelian genetics. Then we separately studied about the non-Mendelian genetics. What will come under the non-Mendelian genetics? Incomplete dominance, codominance, multiple alleles. Okay, in the multiple alleles concept, that is also coming under the non-Mendelian genetics, and then pleiotropy, polygenic inheritance. So in this way, these concepts, all these will come under non-Mendelian genetics. They will not follow Mendel's principles of inheritance. So that's why here they are talking about the polygenic trait inheritance. Will it follow Mendelian laws, Mendelian principles? No, it will come under non-Mendelian inheritance pattern. Clear? This is what the answer is. Next one, again from the principles of inheritance and variation. So here they have given a pedigree, okay, and based on this pedigree, so here they are asking about the F3 generation. In the F3 generation, what they are expecting? They want a normal disease. Means normal means what? There is no disease. Normal individual, but it should be carrier. Likewise, what is the probability? What is the probability that an individual having no disease but being a carrier? So this is what the question in F3 generation they are asking. Okay, this is what the question they have asked. The first thing, the first major problem with respect to this particular pedigree is with respect to this particular symbol. They have given a circle and then there is a dot. But in the NCERT textbook, if you see, they have not used this particular symbol. But here directly they have used this particular symbol. Yes or no? Nice. Next one. Generally, whenever we are teaching about this particular concept with respect to this recessive disorders, there are some symbols that are used for the carriers. First, I will tell you the basic point, then you will understand this. Okay. So generally, when we are talking about this particular carriers, generally we use some symbols. This is the circle means what? Female. This is the half-filled circle. Half-filled circle. And then there is a square, and for this also half-filled circle, half-filled square, and then there is a circle with dot. There is a circle with dot. Generally, these symbols also we are using. Generally, when we are doing the practice, we are using these symbols. And these two we are using with respect to autosomal ratio. Yes, sir. And generally, this symbol we are using with respect to X-linked ratio. X-linked ratio. Understood or not? In this way, by using these particular standard things, generally we are doing this pedigree analysis. Yes or no? So now the major and important question is, in this given pedigree, the same pedigree I have drawn here. In this given pedigree, they have given a circle with a dot. So what you need to do here? So do you need to consider this as X-linked ratio? Do you need to solve it based on the X-linked ratio? No. No. Please remember this. So this is why I am repeatedly telling you people to go through NCERT QR code content. NCERT QR code content. So what is this NCERT QR code content? Now let us see this. This is what the question. I will give you the answer separately. First we will see about the NCERT QR code content. So if you scan the QR code that was given in the NCERT textbook, so now you will get some links, and in those links they have given some extra information. Okay, from the beginning of the year, I am telling you to go through this content also. Since that QR code is printed in the NCERT textbook, so they can ask in the NEET examination, they can ask questions from that QR code content also. And now it is the proof that they are asking from the QR code content also. Yes or no? Now let us see this. And here, this is the NCERT. See here, NCERT not to be republished. Yes or no? I hope you can understand. This is a soft copy. Okay. Now, in the NCERT, they have given this symbol: normal female, normal male, male, affected female. We know these symbols. But what about this? Circle with dot or circle, it is crossed like this. And this they are calling as carrier female. And this carrier female, these symbols you can use for autosomal ratio, you can use for X-linked ratio also. These are general symbols. Similar way here, square having dot at the center, or square that is cutting horizontally here. Now these are used as symbols for carrier male. And where they have used these symbols? In the NCERT itself. And the same symbol they have used here. And not only this, anyhow, these are some normal things. And now in the same PDF, of NCERT QR code content, this is the example they have given. Some practice example they have given. What is the heading? Autosomal recessive trait. In the autosomal recessive trait, to say that it is a carrier. So this is autosomal recessive disease or trait, carrier female. Understood or not? So they have used here carrier female, carrier male. With respect to autosomal recessive also they have used. And even with respect to X-linked recessive also they have used these symbols. Understood or not? So that's why now here the point that you need to understand before solving that pedigree, the point we need to understand is this symbol represents carrier. But anyhow, they have that also they have mentioned, the question itself they have mentioned. But which carrier you need to take, autosomal recessive or X-linked recessive? In both of these you can take. In both of these you can take. Now come to the solving. If you can take it for both of these, and now let us solve it. And when we are solving this, first let us consider that if it is autosomal recessive, please remember carriers are present only in recessive disorders. Carriers are never present in dominant disorders. So you don't need to check this for autosomal dominant and X-linked dominant. Carriers are present only in recessive disorders. So you need to check for autosomal recessive and then you need to check for X-linked recessive. Here we will check for autosomal recessive and it's the same pedigree it is. And here we will check for X-linked recessive. Both of them we will see. And then we will go with the options. And then which one is the better one? Better one only we need to select. Okay. Now let us see. Now let us assume that this pedigree is following autosomal recessive. Then how you will solve this? This is a normal individual. Autosomal recessive normal individual means what? It is capital A, or it is capital A, small a. Yes or no? It can be capital A, small a. When both of them are possible, what I told you? Take heterozygous. When you are getting both the options, go for the heterozygous. Now this is the disease individual. Small a, small a. So now from this, are these possible or not? Carriers, yes. From this capital A, from this small a, possible or not? Yes. Here also same, capital A, small a, carriers are possible. And here this individual is normal, means capital A or capital A, small a. When both of them are possible, you can go for heterozygous. Here also the same, heterozygous. Understood or not? Now the next generation. Here what they give carriers. Is this possible or not? Capital A, small a, yes, possible. Possible. Yes or no? And here it is. It is. Capital A, small a you can write. Capital A also you can write. Anything you can write. No problem. Capital A, small. Anything you can write. You don't think that they have not given this dot means it should be homozygous dominant only. That you cannot say here. Okay. And now here they have given the disease male. Is it possible or not? Small a from this, small a from this. Yes, it is possible. Yes or no? And now this is important. Now focus on this. Now focus on this particular one. This is very important for us. This female genotype is capital A, small a, and this male genotype is small a, small a. And now here what are possible? Now let us see this. When one is capital A, small a, and other individual is small a, small a, what are possible here? Now here you just write it. Here capital A, small a, small a, small a. 50%, 50%. Yes or no? Capital A, small a is normal, phenotypically normal. Yes or no? But small a is phenotypically disease containing. Disease containing. And what is this normal? Is it 100% means normal means is it homozygous normal or heterozygous? It is heterozygous. Since it is heterozygous, can you call this as carrier? Yes, carrier. Okay. And the same concept is applicable for both males and females. Males or females, whatever it is. So in the population, what is the probability here? There is a 50% chance, 50% chance means what? Probability is 1/2. There is a 50% chance that the individual is normal. Normal means what? Not having disease, but carrying an allele that is able to cause a disease. That means carrier. Phenotypically not having disease, but genotypically heterozygous, carrying a disease causally. This is what actually they are asking in the question. In the F3 generation, here, what is the probability that the individual will be not having disease, but carrying disease causing allele? So what is the probability here? 50%. 50% means what? 1/2. So what is the answer here? 1/2. This is what the answer they are asking. So, anyhow in the options 1/2 is there. 1/2 is the answer. Clear? But this you are getting if you are following this autosomal recessive. Now you may have the doubt. Suppose you are following X-linked recessive. Now let us see. Suppose you are following X-linked recessive. Now with respect to X-linked recessive, we will write. And here what I will do is, for the X-linked recessive, to avoid the confusion, you can write anyhow. You can use capital X, small x, that combination you can use. But what I will do is, sometimes while writing capital X, small x, after some time, if you are not clearly writing, you will be confused again. So that's what I will do is, I will use X plus. X plus means dominant X and normal X is recessive X. That will be, there is no confusion at all. So please remember, I will be using X plus as dominant and X as a recessive. Okay. This is how I am using. X plus is dominant over X. So now let us write. And male means what? Definitely one Y chromosome will be there. And normal means what? X plus. Please remember, this is recessive disorder. Recessive allele will cause the disease. So male normal means it should be X plus Y. And here female is having the disease. In X-linked recessive, female is having disease means what? Both of them are recessive X only. So that's why it is XX. I hope you understood this. And now from this, is it possible or not? Is the carrier female possible or not? Yes. X plus from father and X from mother. Same thing here also. X plus from father, X from mother. So carrier female is possible here. Clear? And here male from outside. So outside male. Y chromosome. Normal means what? X plus. And here it is Y chromosome. X plus Y. Understood or not? Clear. And now next generation. Is it possible or not? Now let us check it. Now this female possible or not? Yes. X plus from here, X from here. Possible. Same way here. X plus X. Possible. And here, is it possible or not? Normal female. Okay. You can consider this as X plus X. Carrier also you can write. Or X plus also you can write. Both of them you can write. No problem. Okay. But I am writing this one to avoid the confusion. Right. Now our focus is this one. Is this disease male possible or not? So Y chromosome from the father. Okay. So X. Disease causing X from the mother. Possible. So this is possible. And now here you are getting this genotype and here you are getting this genotype. And now here you need to write very clearly. Okay. So this will take a little bit more explanation. So in the next slide I will write X plus X. Remember this. This is the X plus X. One individual is X plus X. Married to other individual having XY. Having XY. Now observe carefully. Two types of gametes. X plus and X. Here also two types of gametes. X and Y. And then combine this. X plus X. X plus Y. XX. XY. Okay. First we will write whether they are females or males. Two X chromosomes, so female. One X, one Y chromosome, it is male. Two X chromosomes female. One X, one Y male. And now whether they are normal or disease having, containing disease. Now here heterozygous, so it is normal, but it is carrier. Normal but carrier. Understood or not? Next one here, X plus Y. X plus Y. So here it is X plus is there, so dominant is there, so it is normal. And here both of them are recessive, so it will cause the disease. This is the disease condition. And here XY is again disease condition. Understood or not? And here, what is our question? In the F3 generation, this is the F3 generation information, right? So here, this information we need to use here. Okay. So in this F3 generation, what we need? Normal means not having disease. Not having disease means forget about these two. Not having disease. In the not having disease also what we need? One disease causing allele should be there. That means they are asking for the carrier. What is the chance of carrier here? It is 1/4. What is the chance of carrier here? It is 1/4. But the question is, can you use this directly 1/4 in the option? In the option, if you see 1/2 is there, 1/4 is also there. Yes or no? But if you clearly observe the difference between both of these things, here this in the auto, if you are considering as autosomal recessive, what happens? These two are male individuals, right? This is the male. This is the male. This is the female. This is also female, right? Symbol, based on the symbol, you can guess it. Squares are male, circles are female. Whether it is a male or female, whatever it is, the chance of carrier is 50%. So with respect to male, if you see 50%, With respect to female, if you see, it is 50%. Even for the entire progeny, if you see, still it is 50%. So 50% means 1 by 2. That answer 1 by 2 is applicable if you see individually or combinedly. Anyhow, you just see that 1 by 2 is correct here. There is no doubt here. But here, if you are considering as X-linked ratio, then what is the problem? Now observe carefully. Here the problem is you cannot directly say that since you wrote like this, you are getting 1 by 4. So answer is 1 by 4. No, you cannot write like this, because in this case, what you need to observe carefully. I'm using a different color here. With respect to males, you just see male and male. Are you observing carriers in the male? No, there are no carriers for the male. There are no carriers for the male. So here carrier you are observing only for the female, and that too in the female only. See here, this is female, carrier female. So what is the percentage chance of carrier female? 50%. So with respect to males, now observe carefully. With respect to males, what is the chance of carrier? I am writing here. Chance of carrier here is 0%. With respect to male, it is 0%. It is not 50-50. Please remember. With respect to male, chance of becoming carrier is 0%. But now the question is here. The chance of this female to become carrier is 50%. Understood or not? And the chance of this female to become carrier is 50%. Here 50% chance, here 50% chance. The point here is, when you are observing the overall chances, what happens? Both of them can be disease conditions. Yes or no? When you are getting all these possibilities, combine both of them. Then what are the possibilities you will get? Both of them can have the disease. Both of them can be carriers. First one disease, second one carrier. First one carrier, second one disease. So total of 4 options you are getting here. Understood or not? So based on the 4 options, 4 different possibilities you will get. Do you able to understand or not? Anyhow, with respect to me, leave it. And here, what is the chance of this particular individual? What is the chance of carrier in the F3 generation? In the F3 generation, now you just observe carefully. What is the chance here? Now again, it will create a confusion. Whether if both of them are carriers, different proportion you will get. If one is carrier, other one is not, you will get a different proportion. Yes or no? So in this way here, what happens is, anyhow, simply they are asking about what is the probability. So you cannot simply say 50% here. You cannot simply say 50% here. Please remember here. Because if you say 50% means what? Only one is carrier, other one is disease. How you will say that? Both of them can be carriers also. In that case, what happens? 50% of 50%. Again, it will become 1 by 4. So it will create some problem. And if you are considering only 1 by 2. So in this way, 1 by 2 possibility is coming, 1 by 4 possibility is coming. So in this way, different confusions are there with respect to X-linked ratio. I hope you understood this. So with respect to X-linked ratio, some confusions are there. You cannot arrive for one single option. But if you are considering with respect to autosomal ratio, whatever is the way you are taking, definitely you will get a single answer. That is 1 by 2. Anyhow, in our options also 1 by 2 is there, right? Anyhow, in our options also, here 1 by 2 is there. So that's why my suggestion is, for this particular pedigree, consider it as autosomal ratio. So using that autosomal ratio, solve the pedigree. So you will get the answer as 1 by 2. Clear? Okay. So if someone is still trying to tell you that this circle with dot is used only for X-linked ratio, if anyone is arguing like this, show them this particular NCERT QR code content PDF. This is the complete. I did not show the complete PDF. It is having so many pages. You can understand this. This is the figure number 11.1 for the QR code content. Okay, and this is the 11.3. This is the 11.6. So it is having a huge content in it. Okay, so go through it. So here they have used the same symbol with respect to autosomal ratio. The same symbol they have used with respect to X-linked ratio also. So you cannot say that it should be used only for X-linked ratio. It can be used for both. So in this given question, with respect to autosomal ratio, if you consider, so it will be easy. So the better option will be 1 by 2. Clear? But still, when NTA is giving the answer key, so let us see what they will think. but I hope now you understood this particular explanation. Clear? So that's how we have solved that particular pedigree analysis question. Next one. So again, from the principles of inheritance and variation, here the standard question, dihybrid cross with respect to this they have given. Parental generation, capital R, capital R, capital Y, capital Y. Other one is small r, small y, small y. In the F2 generation, what is the phenotypic ratio? It is 9:3:3:1. Standard question. Okay. Next one, from the molecular basis of inheritance, huge number of questions they have asked here. So maximum number of genes are present on chromosome number 1, direct question from the NCERT. And here they have given about some scientist and some conditions they have given. What are the explanation? Direct as it is NCERT content. No need to explain here. Answer is option 1. Okay, next one. Which one of the following are the post-transcriptional events in the eukaryote cell? This you need to listen very carefully. Here there is a small confusion here. In the question they are asking about post-transcriptional. What is the meaning of post-transcriptional? Post means after completion of the transcription. What are the events that we are talking about after completion of the transcription? Observe carefully. Poly A tail addition we are talking. Splicing. What is splicing? Removal of introns and attachment of exons we are talking. And then export of mature mRNA. It is not pre-mRNA. Export of mature mRNA from the nucleus to the cytoplasm through the nuclear pore. These are the things that are happening after transcription. Clear? So anyhow, here is the NCERT diagram. I will explain you. There is one confusion here. So now we will focus on this. Transport of pre-mRNA to cytoplasm prior to splicing. No, splicing is also occurring in the cytoplasm. Sorry, in the nucleoplasm. Splicing is also occurring in the nucleoplasm. So pre-mRNA is converted into mature mRNA. So mature mRNA only will be exported. So this statement is wrong. Transport of pre-mRNA to cytoplasm is wrong. It is wrong. Okay, remove it. Next one. Removal of introns and joining of exons. Is it post-transcriptional? Yes. After completion of the transcription, see here, mRNA is released. Though they are saying mRNA, it is the pre-mRNA. You can also call it as hnRNA, okay? Or precursor for mRNA, anything you can say. So once it is released, then it is undergoing the splicing. In the splicing, introns are removed and exons are attached. So this statement is correct. So removal of introns and joining of the exons is correct. Next one, B is correct. Next one, addition of methyl group at 5' end of hnRNA. Now observe each and every point. Addition of methyl group at 5' end of hnRNA. Is it a methyl group? Not just methyl group. It is not just methyl group. It is 5-methyl. So it is 7-methyl. 7-methyl guanosine triphosphate. It is not just methyl group. It is the 7-methyl guanosine triphosphate. It is in the NCERT they have not given this much name. Simply they said an unusual base will be added. That's what they said. But actually it is the methyl guanosine triphosphate. But still okay. Okay, let us consider that it is the methyl group. No problem. But still there is another problem. What is that problem? Addition of methyl group at 5' end of hnRNA. And this is C. Post-transcriptional they are saying. After transcription, is this molecule added at the 5' end? And this is C. If you see C was given everywhere. C was given everywhere. That means as per these options, C is the correct statement. But actually it is a wrong statement. C is actually wrong statement because this cap methyl guanosine triphosphate attachment at 5' end is, see, even before completion of the transcription, still transcription is taking place, right? So it is actually a co-transcriptional, co-transcriptional process. It is not post-transcriptional process. Please remember this. Anyhow, here in all the options C is there. So you don't need to worry. You need to consider, definitely you need to consider this as correct statement. But I am telling you the concept. Actually, this addition of the 7-methyl guanosine triphosphate is not a post-transcriptional. It is a co-transcriptional. During transcription itself, it is added. Okay. But anyhow, actually this should be eliminated. But anyhow, all the options are having. So we cannot do. So that's why B and C are correct. Next one. Addition of adenine residues at 3' end of hnRNA. Technically, at this level of class 11th and 12th, you don't need to worry. But technically, if you say, you should not call this as adenine residues. You should call them as adenylic acid or adenylate. Adenylate residues are added because it is not just adenine that is added. It is the adenine plus ribose plus phosphate. Yes or no? incoming are adenosine triphosphates, and when they are making the bond, two phosphates will be removed, so single phosphate will be there, so that it will become adenosine monophosphate. You can also call it as adenylic acid. So you should technically, if you're speaking, addition of adenylate residues at 3' end is more better, but still okay. This is still, at least they gave adenyl residues. Let us consider this as correct. So now what happens? B, C, D, these three are correct. Clear. Next one, base pairing of two complementary RNAs. No, from one segment, that means from one transcription unit, only one strand will act as template, only single mRNA will be produced. That's why there is no complementary RNA. So A and E are wrong. So B, C, D are correct. So as per the options, if you are going, you need to select 3. So which 3 you need to select? B, C, D only you need to select. So B, C, D only. Okay. In this way, this is the answer. You are forced to select this particular option. Clear? Even though there are some small mistakes are there, still this is the better option as per the options. Okay, nice. I hope you understood this particular question. Next one, again from the molecular basis of inheritance. They have given two statements. First one, in the RNA world, RNA is considered the first genetic material evolved to carry out essential life process. Correct. RNA acts as the genetic material and also as a catalyst for some important biochemical reactions in living systems. Correct. Being reactive, RNA is unstable. So all these things, 3 points they have given, all the 3 are correct for statement 1. Next one, statement 2. DNA evolved from RNA and is more stable genetic material. Correct. So due to small modifications from in the RNA, so DNA got evolved. So it is correct. It's a double helical strands being complementary, resist changes by evolving repairing mechanism. All these things are correct here. So both statement 1 and 2 are, both of them are correct. Clear? Next one, again from molecular basis of inheritance, statement 1 and 2. Statement 1, transfer RNAs and ribosomal RNAs do not interact with mRNA. This is wrong. They are interacting with mRNA. Yes or no? So during the translation process, they are interacting here. So that's why it is wrong statement. Next one, RNA interference, RNAi takes place in all eukaryotic organisms as a method of cellular defense. In that biotechnology applications, this statement was given. So it is correct statement. So 1 is wrong, statement 2 is correct. So this is what the answer is. Next one, who proposed that genetic code for the amino acids? The genetic code that is coding for amino acids should be made up of 3 nucleotides. Who proposed this? In NCERT very clearly they go, who proposed? It is not about proved, it is proposed. Who proposed? It is the George Gamma. Yes or no? Next one, histones are enriched with. So histones are enriched with what? Basic amino acids like lysine and arginine. Very clear sentence from NCERT. Okay, next one. So which factor is important for termination of transcription? In the case of prokaryotes, for the transcription termination, what is the factor that is required? Rho factor is required. See what the question they have asked. Next one, from the evolution chapter. Sweet potato and the normal potato. Okay, and these two represent a type of evolution. What are these? So here, with respect to sweet potato, if you see, it is the root modification. With respect to potato, if you see, it is a stem modification. But both of them are underground modifications, and they are showing some similarity. But origin wise, if you see, one is root, another one is stem. So that's origin wise, they are having different origins. So what type of evolutionary terms are related to this one? So they are showing analogy. Yes or no? They are examples for analogous structures, and they are showing convergent evolution. Origin wise different, but they are showing similar type of structure. So that's why it will come under convergent evolution. So analogy and convergent evolution. This is what the answer is. Next one, from the human health and disease. From this also huge number of questions they have asked. Next one, neoplastic characteristics of the cells. Neoplastic here, please remember, with respect to the malignant tumors they are talking. In NCERT very clearly they have given this particular sentence. Okay. A mass of proliferating cell. Yes, this is the general property of the tumor cells. Next one, rapid growth of the cells. This also we are observing. Next one, invasion and damage to the surrounding tissues. Yes, this is what the property of malignant tumors. But the last one, those confined to original location. This is what with respect to benign, but these neoplastic cells. So we are talking with respect to malignant. So that's why what are correct? A, B, C statements are only correct. Okay, next one. Next one, again they have given some statements. We need to identify what are correct. So CT, computed tomography, and MRI, magnetic resonance imaging, they detect cancers of internal organs. Direct statement from NCERT. This is correct. Next one, chemotherapeutic drugs are used to kill cancer or non-cancerous? Cancer cells, but here they gave non-cancerous cells. So B statement B is wrong. Next one, alpha interferon activates the cancer patient's immune system and helps in destroying the tumor. True or false? It is true. So A and B, C are correct. B is wrong. Next one, D. Chemotherapeutic drugs are the biological response modifier. No, in NCERT very clearly they give alpha interferons are the biological response modifiers, not the chemotherapeutic drugs. So D is wrong. Next one, E. In the case of leukemia, blood cell count increases or decreases? Leukemia, blood cancer count will be increased, but here they give decreased. So here they are asking about correct, no? So what are correct? A is correct and C is correct. That's it. Only two are correct. A and C are correct. Okay. Next one. Again from the human health and diseases. After maturation in the primary lymphoid organs, the lymphocytes migrate for interaction with the antigens. So where they are going? They are going to secondary lymphoid organs. So from the given list, you need to identify secondary lymphoid organs or secondary lymphoid tissues you need to identify. Thymus and bone marrow are primary lymphoid organs. So remove this. Spleen, secondary lymphoid organ. Lymph nodes, secondary lymphoid organ. Peyer's patch, secondary lymphoid tissue. Peyer's patch, galt, malt, all these are examples for this secondary lymphoid tissues, right? So what will come under secondary lymphoid organs or tissues? C, D, E. These will come. So answer is option 2. Okay. Next one. Which of the following type of immunity is present at the time of birth and is a non-specific type of defense in the human body? So non-specific but present by birth. What is that? Innate immunity. Okay. Next one. Identify the statement that is not correct. So first you need to observe the NCERT diagram here. This is the antibody structure. In the antibody structure, there are two heavy chains and there are two light chains are present. And these are protein chains. Since they are the protein chains, observe this. This is the N terminus. This is the C terminus for the heavy chain. For light chain also, this is N terminus and this is the C terminus. Clear? Next one. Not only this, in these chains, there are some regions called as constant regions and then variable regions. On which side the variable regions are present? Heavy chain, light chain. For both of them, towards the N terminus side, variable region is there. Towards the C terminus side, constant regions are present. Same thing here also. Towards the N terminus side, variable region is there. And towards the C terminus side, constant regions are present. And two heavy chains, two light chains are present. And they are connected with each other by disulfide bonds. This is the basic information. And based on this, they have given some statements. We need to identify which one is not correct, which one is wrong. We need to identify. Antigen binding site is located at C terminus. This is the antigen binding site. Is it located at C terminus site or N terminus site? N terminus site. But here they are saying that it is located towards the C terminal region of the antibody molecule. So it is wrong statement. Remaining all are correct, but still let us read it. Constant region of heavy and light chains are located at the C terminus of antibody molecule. For this constant, this is the constant located towards C terminus. For this constant is this one, it is located towards C terminus. So it is correct statement. Each antibody has two light and two heavy chains, correct? And the heavy and light chains are held together by disulfide bonds. See, disulfide. So in this way, they are connected by disulfide bonds. So in this way, option 1 is the not correct statement. Clear? Next one. From the microbes in human welfare. Generally, one question is asked from this particular chapter. But here they have asked total 4 questions. Let us go through this. So which of the following is an example of non-distilled alcoholic beverages? So generally in the yeast fermentation, the maximum percentage of alcohol that is produced is 12 to 15%. Beyond that, it cannot be produced. So if you want more percentage of alcohol, it should be produced by distillation process only. So rum, whiskey, brandy, these are produced by the distillation. But in the question they are asking about non-distilled, directly from the fermentation you can get it. What are these? The beer, wine, these are the examples given in NCERT that are non-distilled alcoholic beverages. So answer is beer. Okay. Next one, another example, another question from the microbes in human welfare. They have given some list of organisms and they are asking about which one cannot fix the atmospheric nitrogen. So in this particular chapter, in the last section, they are talking about biofertilizers. So in that biofertilizers, so some different organisms are having the ability to fix atmospheric nitrogen. Likewise, we have studied. So in that list, we studied about Azotobacter, Oscillatoria, Anabaena, Nostoc. These examples we studied. But Volvox, no, we have not studied. At least based on the elimination also you can easily understand this. So Volvox cannot fix the atmospheric nitrogen. So answer is D, Volvox only. Clear? Next one. Streptokinase enzyme that is produced from the bacterium Streptococcus. What is the use of this? It is used as a clot buster. So it is helpful for dissolution of the clots. So here directly they have given removing the clots from the blood vessels. Direct answer. Next one, which of the following microbe is not involved in the preparation of household products? In the NCRT, different microbes names they have given under different headings. So use under the household products, use for the industry level, use for the acids production, some chemicals production in the detergent industry, like with different headings they have given, right? So based on that, you need to observe this. Aspergillus niger, that is used for the production of citric acid production. This is used in the industry level. For the industrial purpose we are using, but not for the normal household purpose. So this is not used. So please remember this. Lactobacillus we are using for curd preparation and all. Trichoderma polysporum. So that different chemicals we are producing, right? Bioactive agents we are producing. So this is a different thing. Next one, Saccharomyces cerevisiae. This we are using for bread production and all. And Propionibacterium shermani, that cheese production and all. So here what are not related to this use, what are the microbes that are not related to household uses? Aspergillus niger and Trichoderma polysporum, these two, A and C. So this is option 4. Clear. Next one, from the biotechnology first chapter, here also huge number of questions they have asked. Even some application questions also they have asked here. So some students felt a little bit difficult, but if you are conceptually strong, you can easily answer these questions also. Okay, now let's quickly go through these questions. Here they have given a diagram. So here origin, this is a plasmid, origin of replication they have given, and two selectable markers they gave, TATR, tetracycline they gave AMP, but actually it is AMP-R, ampicillin resistant gene. And here they have mentioned about BAMH1 site. And not only this, they have given another marker, that is beta galactosidase enzyme producing gene, and within that gene there is a site called as EcoR1 site. And in the question they are saying that if alien DNA or gene of interest, if it is introduced at the EcoR1 site, if it is introduced at EcoR1 site, then how you will identify the recombinants? So for this, first we need to understand the basic concept, so that I will explain, and then we will come back to these particular options here. Okay, so this one completed, this one also completed. Yes, this is what the diagram that they have given in the NCERT. Okay, so let me use a different color so that it will be easy for you. So this is what the diagram that they have given. They are saying that you need to introduce gene of interest at EcoR1 site. Now here you will be getting two options. If gene of interest is not inserted, now it will be the original plasmid. Okay. And if gene of interest is inserted at the EcoR1 site, what happens? This EcoR1 site will be split, and in between this gene of interest will be introduced. So that what happens to the original beta galactosidase enzyme gene? It will be broken, it will be split. Yes or no? Because we are inserting our gene of interest. So in this way, insertional inactivation of beta galactosidase gene will take place. Understood or not? So that will be, that means that being laxate, beta galactosidase producing is nothing but the laxate. So that will be inactivated. That's why here I have not mentioned, because due to insertion of this, it got broken, it got separated. So now it is not able to produce beta galactosidase. Now, if this is the condition, in simple, I am trying to give you the answer. So observe this carefully. If this is the condition, what happens? In this condition, that means here recombinant is not formed. That means it will come under non-recombinant. So here what happens to beta galactosidase? Beta galactosidase is produced. It is produced. Okay. But here what happens? The gene for beta galactosidase production got split. It got broken by the insertion of gene of interest. So that's why here what happens? When the gene is broken, it cannot produce beta galactosidase. So here beta galactosidase enzyme is not produced. This is the basic difference you need to understand. And the next point you need to understand is, here we are using some substrate. Same concept is useful to answer one more question. In the next coming, in the same biotechnology only, another question they have asked about blue-white colony screening. This is blue-white colony screening only, so listen very carefully. So here we are adding a substrate. In this concept, we are taking a substrate. What is the color of this substrate? It is called as a chromogenic substrate. Chroma means color, genic means producing. Color producing substrate, but it is not having color. It is colorless. It is colorless. And standard example is X-gal is the substrate. Okay. And this substrate is called as chromogenic substrate because it is a color producing. Does it has any color? No, but it can produce color. That means who will have the color? Product is having the color. Product is having the color. What is the color of this product? Blue color product. Okay, please remember the meaning of this particular sentence. Very clearly I have explained this. So substrate is colorless, product will be having the color. So that's why this substrate is called as chromogenic, color producing substrate, but substrate is not having any color. This is what. But when it can be converted, from colorless substrate to colored product, if there is a beta galactosidase enzyme. If beta galactosidase enzyme is produced, then only this colorless substrate will be converted into blue color product. So now what is the overall concept here? This is the non-recombinant. This is non-recombinant. This is non-recombinant. This non-recombinant is able to produce beta galactosidase. That's why it is producing blue color colonies. So what is the overall conclusion? So non-recombinant will be having this blue color. Bacterial colonies which are having non-recombinant bacteria, now they will appear blue in color. Then what about these things? This gene of interest is inserted, so that's why these are called as recombinants. These are called as recombinants. Yes or no? In the case of recombinants, the beta galactosidase producing gene is inactivated. So it cannot produce beta galactosidase. If beta galactosidase is not produced, if enzyme is not there, substrate cannot be produced, becoming product. Yes or no? Substrate cannot be converted to product. So substrate will be like that only. So like that only means what? Colorless. It is colorless, but these E. coli bacteria, generally we are talking with respect to E. coli bacteria, and these E. coli bacteria are growing on LB medium, Luria bertini medium. On the LB medium, the original color of these E. coli bacteria is white color. Original color of the bacteria is white color. On this white color, you just imagine a colorless. Colorless means what? Transparent. Transparent. Suppose this is the black. Suppose this is the black. On this black, I am keeping one transparent sheet. What you will see? Black only you will see. Same way, originally the colony is white in color. White in color. Substrate is colorless, transparent. So what you will see? Original white color only you will see. So that's why these recombinants, they will appear white in color. White in color. So this is what the concept is, okay? But if you still have the doubt, anyhow, in the regular class, I have very clearly explained. Since it is just an explanation, in short, I am concluding this. So in this way, non-recombinants will be having blue color colonies, whereas the recombinants will be having white color colony. Is the concept clear? Same concept. Please remember, once again, it will be useful when we are solving another question. Okay. So here, that same logic they are saying. If gene of interest is inserted, EcoR1, then which one of the following strategies will be chosen to select the recombinant colonies? With respect to recombinant, they are asking. So which color? White color colonies. White color colonies will be selected. Yes. If you want recombinants, you need to select white color colonies, not the blue color colonies. Next one. Anyhow, answer is the option one. Next, blue colored colonies grown on ampicillin plates. No, they are non-recombinants. We don't want them. Next one, using ampicillin tetracycline containing medium plate. So please remember here. Ampicillin tetracycline resistance they are talking. This is also very important concept. Observe carefully. This is not the as per NCERT concept. So this is a little bit slightly application they have given. See, this is non-recombinant. This is recombinant. But if you clearly observe, in non-recombinant, ampicillin resistant gene is normal, tetracycline resistant gene is normal. Even in the recombinant also ampicillin gene, resistant gene and tetracycline resistant gene is normal. So in both the cases, both the genes are normal. They are not inactivated. So these non-recombinants can grow on ampicillin and tetracycline. Recombinants also can grow on the same thing. So simply based on the ampicillin and tetracycline, you cannot select these because both of them can grow on both the antibiotics. So that's why that option is also not there. So this is not useful. Next one, blue color colonies will be selected. No, we don't want blue. We want only white color colonies. So option one is the correct. Clear? Next one. Another question. Polymerase chain reaction amplifies DNA following the equation. So this equation very clearly in the regular class we have discussed. Okay, here actually there are two formulae are there. What are these two formulae? So what are these formulae? So here there are two formulae. First one is for the total DNA molecules in the PCR process. total DNA molecules. If you want the total DNA molecules count, what is the formula that you need to use? 2 to the power n. Okay. And if you want target DNA molecules, there is a small difference between these two. If you want target DNA molecules count, target means what? Whatever is the, from starting with the forward primer until the reverse primer, whatever is this target is there, only the target molecules. If you want only the target, not the other, not only this, some extra molecules also you are getting. In the regular class we have discussed. So if you don't want them, if you want only the target DNA molecule, that is the formula. But here in our question, what they have asked? They are asking about total DNA molecules. So if after n number of cycles, how many DNA molecules will be produced? 2 to the power n. After n number of cycles, how many targeted DNA molecules are produced? 2 to the power n, 2 to the power n whole minus 2n. These are the formula. Very clearly in the regular classes we have discussed. And now this is what the question they have asked in this NEET examination. So what is the answer? It is the 2 to the power n. That's what they are asked. Clear? Next one. Identify the part of the bioreactor. So labeling they have given here. So foam breaker. Where is the foam breaker? Please remember here. Here we are using the liquid culture medium. Liquid culture medium. So that's why in the labeling, if you see, they have given in NCERT that broth, liquid broth they have mentioned. So when this broth, when we are actually vigorously mixing with the help of this impeller blades, what happens? Foam will be generated, and this foam will be accumulated at the top space. There is a space we are deliberately we are keeping some space here for the accumulation of the foam, and that foam will be accumulated here. And this accumulated foam should be removed. What is the reason why it should be removed? All the things in the regular class we have discussed. So please remember, so at this top portion, foam breaker, so they will break the foam, so that they are trying to reduce the foam, remove the foam, and these are the things that are present. So these are the foam breakers. So the labeling, with respect to labeling, if you see which one represents the foam breaker, it is the C. So that's why labeling is also very important. So all the labeling with respect to NCERT diagrams, so you should remember. Okay, next one from the same chapter. Next one, another question related to blue-white colony screening. So observe carefully. The blue and white selected markers have been developed which differentiate recombinant colonies from non-recombinant colonies on the basis of their ability to produce color in the presence of chromogenic substrate. Same concept once again they are saying here. Okay, how exactly we are using? They are saying here. So given below are the two statements. They gave two statements. We need to identify which one is correct or wrong, like this. Statement 1. The blue colored colonies have DNA insert in the plasmid and they are identified as recombinant colonies. So if they are recombinant, so insert is present, and if they are recombinants, do they produce blue colony? No. If gene of interest is present there, no, they will not be producing blue color and they will be colorless. That means statement 1 is wrong. Just now I gave the explanation, right? Next one, statement 2. The colonies without blue color means white colonies. That means white colonies have DNA insert, correct? White colonies, why they are white? Because DNA insert is present. That's why insertion inactivation is taking place and in the plasmid and are identified as recombinant colonies. This is correct. So statement 1 is wrong and statement 2 is correct. I hope same explanation. Okay. So now next one, another question. So which of the following enzymes are not essential for the gene cloning? And here some of these names they have not mentioned in NCERT, but based on the elimination process you can easily identify this. Based on the options, if you see, you need to eliminate two enzymes. Okay. Restriction enzymes, they are compulsory. DNA ligase for the attachment, it is compulsory. DNA mutase, you have not studied this enzyme, so this is not required here. Like that also you can remember this. This is not required. DNA recombinase is not required here. Okay, so this is also not required. DNA polymerase in the PCR you are using. So A, B and E we are directly using as per the NCERT content, right? And C and D you are not using. So these are the things that are not required. Directly you can write CD, only CD. Okay, direct answers. Next one. Another question from the same chapter. So they have given some two statements. The DNA fragments extracted from gel electrophoresis can be used in construction of recombinant DNA. Yes, you can use it. That's why we are using the agrogel electrophoresis and all. Statement 2. Smaller size DNA fragments are observed near anode. Yes. Small molecules are moving fast. Yes or no? So that's why they are present nearer to the anode. And while larger fragments are found nearer to the wells in the agrogel. Correct. So both the statements are correct. Yes or no? Next one. So which one of the following genetically engineered organisms are used by Eli Lilly company to prepare human insulin? So E. coli bacterium they have used. So E. coli they used means bacterium. So answer is the bacterium. Next one, silencing. These questions are from the biotechnology applications. Observe carefully. Silencing of specific mRNA is possible via RNAi because of what? Complementary double strand RNA. Even in previous NEET examination also the same question they have asked. Next one, why can't insulin be given orally to diabetic patients? This is the in-text question. Direct in-text question they have given. Why we cannot take insulin orally? Generally, if you see, so we are taking subcutaneously. Insulin we are taking. Why can't you take orally? Because insulin is a protein. If you are taking orally, like all other proteins, this also will be digested. And if this insulin is digested, then what is the use in taking this? So that's why we are not taking orally, because it will be digested in gastrointestinal tract. This is what the answer is. Next one, organism population. Epiphytes that are growing on the mango branch is an example of which of the following? Direct example from the NCERT. It is example for commensalism. And who is known as the father of ecology in India? It is Ramdev Mishra. And this information was given on the ecology unit introduction. So they have given unit introduction page, and just on the next page of this, they have mentioned about the scientist. So that information is also part of NCERT. You need to read for examination. Clear? Next one. So here, though it is from the basic common unit introduction, still I kept it under organism and population. Okay, next one. Another question here. Statement 1. Fig fruit is a non-vegetarian fruit as it encloses the wasps in it. So here it will be like a little bit controversial, but still we consider that it is a correct statement, because so here in this fig fruit, I don't want to explain the complete concept. Here what happens is the male insects, now they will die and they will be present inside the fruit only. So if you are eating that fruit as it is, so now you can consider that yes, it can be considered as a non-vegetarian. Can be, but still it is under conflict only. Next statement 2. Fig wasp and fig tree exhibit the mutual relationship. Yes, direct NCERT statement it is. Okay. So this is a correct statement. As fig wasp completes its life cycle in the fig fruit, and fig fruit gets pollinated by the fig wasp. So it is a correct statement. So statement 1 and 2, both of them we are considering as correct statements. Next one. So another one, they have given about this growth curve and all. So direct formula that they have given in NCERT, answer is option 4. This is directly from the NCERT only. Next one, from the ecosystem. So they have given two statements. The primary source of energy in an ecosystem is solar energy. Correct statement. The rate of production of organic matter during photosynthesis in an ecosystem is called as, what is it called as? Is it called as NPP or GPP? It is called as GPP, gross primary product. It is not NPP. So statement 1 is correct. Statement 2 is incorrect. Clear? Next one. Again, they have given two statements. In ecosystem, there is unidirectional flow of energy of sun from producers to consumers. Yes, there is a sequential flow is there in unidirectional. This is correct statement. Next one. Ecosystems are exempted from second law of thermodynamics. No, they are not exempted. It is a wrong statement. So even ecosystems are also following the second law of thermodynamics. What the second law of thermodynamics says? When energy is moving from one level to the next level, that means when reaction is taking place, what happens? Some amount of energy will be lost into the nature. So that means some disorderliness will be increased, entropy will be increased, yes or no? So these are the some things. So they are applicable even for this ecosystems also. That's why the energy that is present in one level of ecosystem, when it is moving to next level, entire energy cannot go to the next level. Some energy will be lost into the nature. So that's what. So here what happens? Statement 1 is correct. Statement 2 is wrong. Okay. Next one. So what is this formula that they have given? What are the units that they are asking for the productivity of an ecosystem? Direct answer here. Kilocalorie per meter square per year. Okay. Next one. From the biodiversity and conservation. Which one of the following is an example for ex situ conservation? Zoos and botanical gardens. Next one. Here they have given some standard examples from the NCERT. So it is a standard matching. So nothing complicated. Answer is option 4. Clear? Next one. That's it. With this particular discussion is completed. I hope at least, so even though you know majority of the answers for these questions, but for some are definitely tricky questions, so there you may face a little bit difficulty. So and one more thing I wanted to tell you here, even in the previous video also I told you, so when compared with physics and chemistry, yes, biology is a little bit easier, but when compared with biology papers of previous NEET examinations, yes, this time it is difficult. Yes, definitely it is a difficult one. So that's why overall, if you talk about the biology, instead of saying easy, it is very wise to say that it is an average paper, means moderately difficult. It is not too difficult, not too easy. It is a moderately difficult. So this is how they made, but it is a very good paper. So some application questions, some logical, so all the things they have given. It is not too lengthy, but it is a logical paper. Okay, so directly you cannot answer, though you can answer for majority of the questions, but for some of the questions you need to think and then only. Okay, and if you know the concept, then only you can attempt, or else unnecessarily you may lose the marks. It's okay. So this is what, with this particular paper discussion is completed. Thank you so much.